Translation is the process by which proteins are synthesized in the cell after DNA has been transcribed into mRNA. This process is highly complex and involves many intracellular structures. However, the ribosome and the tRNA are of critical importance when it comes to recognizing codon sequences on mature mRNA and identifying the correct amino acid to go in the growing polypeptide chain. To begin the process of translation, tRNA molecules within the cell must bind to the appropriate amino acid. tRNA has a very specific structure that allows it to do this. tRNA has a folded structure with the 3 hairpin loops. Each of these hairpin loops represents an arm of the tRNA molecules. The anticodon arm of a tRNA molecule has a specific sequence of 3 nucleotides that complementarily bind to one unique mRNA codon and identify it. There are 30 to 40 unique tRNA molecules, each of which has a unique anticodon that allows it to accurately identify all possible mRNA codons. On the 3' carboxyl end of the amino acid acceptor arm of the tRNA molecule, there is a conserved CCA sequence. The amino acid that is encoded by the specific complementary mRNA codon to the tRNA anticodon can bind to this carboxyl end of the tRNA.
In order for the appropriate amino acid to bind to the tRNA molecule, the enzyme aminoacyl tRNA synthetase is needed. Aminoacyl tRNA synthetase identifies specific tRNA molecules by a unique RNA sequence on their 5' stem of the amino acid acceptor arm. Once it has identified a tRNA molecule, these enzymes find the appropriate amino acid based on its size, charge, and R group and bring them back to the tRNA, where they catalyze the esterification reaction that allows the amino acid to bind to the 3' end of the tRNA. The carboxyl end of the amino acid is able to bond to the hydroxide group on the adenine and the CCA sequence to form an aminoacyl tRNA. The process of the appropriate amino acid being bound to a tRNA molecule is called tRNA charging. Once a mature mRNA has left the nucleus and entered into the cytosol of the cell, the process of translation can begin.
The process of translation is carried out in ribosomes. Ribosomes are considered ribonucleic proteins because they are comprised of both rRNA and large protein components. The ribosome is split into 2 subunits, the small subunit and the large subunit. The size of the small and large subunits varies between prokaryotes and eukaryotes. In bacteria, the small subunit is called 30S and the large subunit is called 50S. The small subunit of ribosome acts as a binding site for the strand of mRNA, and the large subunit contains 3 unique sites: E for exit site, P for peptidyl site, and A for aminoacyl site. This is where tRNA molecules can bind and then translocate to form the growing polypeptide chain of proteins. Translation is comprised of 3 steps: initiation, elongation, and termination.
For translation to begin, several components are needed: the small and large unit of the ribosome, an mRNA, and a charged initiator tRNA carrying the first amino acid in the protein sequence, usually a methionine amino acid. Translation begins when the initiator tRNA carrying the methionine amino acid binds to the small subunit of the ribosome. Initiation factors like IF3 in prokaryotes help bind the mRNA to the small subunit and prevents the large subunit from attaching to the small subunit. In eukaryotes, the small subunit of the ribosome and the initiator tRNA bind to the 5' end of the mature mRNA at the 5' GTP cap. This complex then reads 30 to 40 sequences of the mRNA until it reaches the start codon, AUG. Because of the complementary nature of the anticodon arm of the tRNA molecule, the initiator tRNA with the anticodon UAC is able to bind to the start codon with the aid of another initiation factor, IF2. IF2 requires a GTP molecule to function. This entire structure forms the initiation complex, composed of the small subunit, the mRNA, the initiation factors, the initiation tRNA, and GTP. Once the initiation complex is formed, IF3 detaches from the complex and the large subunit joins the small subunit. When the large subunit joins the complex, the GTP is hydrolyzed to GDP and is removed from the complex along with all the initiation factors.
Once the initiation complex has been formed, initiation is completed and elongation begins. During this stage, amino acids are joined to create a polypeptide chain. When this begins, the initiator tRNA carrying the methionine is located in the P site of the large subunit. Next to this P site is the A site, where a new codon is exposed. Now, another charged tRNA that contains the appropriate anticodon approaches the A site. Simultaneously, elongation factor 2 joins with GTP and the charged tRNA to form a 3-part complex that enters the A site. Once there, GTP is cleaved off and EF2 is released, allowing the anticodon on the appropriate tRNA to bind to the codon in the A site. Next, the large ribosomal subunit catalyzes the formation of the peptide bond between amino acids that are attracted to the tRNA in the both A and P sites. The formation of this bond allows the amino acids in the P site to be released from its tRNA. The last step is translocation, or the movement of the ribosome down the mRNA in the 5' to 3' direction. With the help of elongation factor G and the hydrolysis of GTP to GDP, the tRNAs in the P and the A sites shift. In this step, tRNA in the P site moves to the E site and the tRNA in the A site moves to the P site. The A site is then left open so that the newly exposed codon can be identified again, and this continues the polypeptide chain.
Elongation ends and termination begins when a ribosome encounters a termination codon. And because tRNA does not contain a complementary anticodon, it leaves a vacancy in the A site. Stop codons can be UAA, UAG, or UGA. The vacancy in the A site allows for release factors to bind to the ribosome and separate the polypeptide chain from the ribosome. Binding of R factors 1 and 2 promote cleavage of the tRNA located in the P site from the polypeptide chain. Next, R factor 3 forms a complex with GDP and produces a conformational change that releases R factors 1 and 2 while causing the tRNA to shift to the E site. After this step, the large and small subunit disengage, releasing the mRNA.
Once the polypeptide has been formed with the appropriate amino acids, the chain may still need to undergo edits before it can become a fully functional protein. However, the cleavage of the polypeptide from the ribosome marks the end of translation. As discussed, the process of protein synthesis involves two important RNA components: the tRNA, which locates and brings specific amino acids to the growing polypeptide chain, and the rRNA component of the ribosome, which allows the mRNA holding the instructions for protein synthesis to associate with the small subunit of the ribosome and be positioned in a way that allows it to be interpreted and then translated. The RNA aspect of each of these components is what allows this process to occur. Since RNA, like DNA, is complementary, only sequences of RNA that contain appropriate anti-parallel nucleotide sequences can associate with one another via hydrogen bonding between the bases. This pairing is highly specific. It is because of this incredibly high level of specificity that allows mRNA codons to be identified by tRNA, allows tRNA to associate with its proper amino acid, and allows mRNA start codons to be recognized by the initiation complex. Additionally, because the genetic code, including codons, is conserved, meaning that one codon sequence will always code for the same amino acid, the high level of specificity that RNA association during translation allows for is the same during every round of translation.